All right. So I'd like to thank the organizers for putting together such a great lineup, and also for them to invite me to tell you about some of our work and give an introduction into heart valve development. So I think without further ado, we'll get started. So starting at the beginning of valves. Okay. So valves start very early in development, in a mouse at about day 10 and a half, in a human about 4 weeks, with the formation of endocardial cushions in the AV canal and the outflow tract. These cushions elongate and then stratify over time in postnatal stages. So the valve progenitor cells arise from an endothelial mesenchymal transition. So cells from the endocardium delaminate, migrate into the endocardial cushions where they proliferate, and later on go on to generate the valves. As the cushions elongate, they begin to synthesize extracellular matrix, proteoglycans, elastin, collagens, and then these matrix components stratify after birth in a pretty poorly understood process, I think, into elastin-rich layer, a spongiosa, proteoglycan-rich layer in the middle, and a collagen-rich layer on the side away from the flow. And so these cells undergo changes over time, and also they undergo diversification in terms of the extracellular matrix that they make. The formation of the cushions and the valve progenitor cells has been studied for a long time, and a lot is known about some of the molecular hierarchies that control the endothelial mesenchymal transition, and then also control the sort of the expansion and proliferation of the valve progenitor cells. So the initiation of endocardial cushion starts with BMP2 expression in the myocardium that then signals to the endothelial cells, and then through multiple signaling pathways, TGF-beta, Notch, Wnt, beta-catenin, and VEGF, are all involved in some aspect of proliferation, delamination, migration of the endothelial cells into the cushions where they form the progenitors of the valves. And within the valve progenitors, excuse me, there are transcription factors that govern the proliferation and the expansion of this population to basically generate the building blocks of the valves that will go on later in development. And an interesting aspect of this is that many of these events are recapitulated during valve development. And so this is histology of human diseased valves. In the control valves, so we're looking for brown nuclei, there's very little expression of these transcription factors. But during both pediatric and adult valve disease, TWIST1, SOX9, which is another transcription factor involved in early valve development, MSX2, these are all activated, and they're activated in a very widespread way. We don't know how they're activated. We don't know what they're doing. We don't know if it's involved in repair or if it's involved in the pathogenesis. But really, it's very striking how these early events in valve development are recapitulated in disease. And I think this is a very interesting area to focus on. And then this is just another view of this, where TWIST1 is a transcription factor. And from our developmental studies, we know that TWIST1 promotes cell proliferation, and it also regulates sort of a primitive valve matrix in terms of collagen 2A expression. And here you can really see that TWIST1 in green and phosphohistone, where the cell proliferation is, are both highly active in adult disease. And again, we don't know what's controlling the initiation of this or what the function of this is in valve disease. So another question that we had is, does an endothelial mesenchymal transition contribute to adult valve disease? And so what I told you was those endocardial cushions form initially through this endothelial mesenchymal transition, where endocardial cells delaminate and then migrate into the cushions. We don't know if that happens in adult disease, and we wanted to look at that more. So the model that we used was an osteogenesis imperfecta model, and this is a mouse model of a bone condition, but these mice also have valve disease, where in the aortic valves they have very thickened and proteoglycan-rich valves, The initial mutation in this is a COL1A2 structural mutation, so their collagen is compromised structurally. It's there, but it's not right. And over time, these valves develop this proteoglycan-rich, thickened phenotype in the aortic valve. So again, going back to what we know about endocardial Cushing's, we know that TGF-beta signaling can promote expression of slug and snail, which are transcription factors involved in cell migration, and that these then go on to, these cells go on to express TWIST1. And we do RNA analysis, we can see that snail1 and snail2, which is also called slug, are both increased in the osteogenesis imperfecta valves. And we also knew from previous studies that the TWIST1 is increased. So again, in this model, we see increased expression of the valve progenitor markers and also these transcription factors that are involved in endothelial mesenchymal transitions. And by antibody staining, I'm not sure you can see this, but we look for phosphosmad2,3, which is an indicator of TGF-beta signaling, and also smooth muscle alpha actin, which is smooth muscle but also activated myofibroblasts. And in the wild type valves, we don't see very much expression, but in the osteogenesis imperfecta, the thickened aortic valves, we can see the pink staining, which is the SMAD2,3, and there's also some indicators of fibroblast activation with the SMA. And then we also looked at this plasminogen activator inhibitor, which is a direct downstream target of TGF-beta, and it's also increased in the knockout, or in the osteogenesis imperfecta. And so these are both indicators that there's increased TGF-beta signaling in these proteoglycan-rich valves. And then when we looked at snail bioimmunostaining, so again, snail's going to be in red. We see very little in the wild-type valves, but in the mutant valves, we see staining on the flow side and also at the tip of the valves indicated in pink again, and this is in areas where we see some cellular disarray as well. And so we think that there may be some hemodynamic or mechanical forces that are involved in activating this endothelium that could be involved in inducing an endothelial mesenchymal transition in these cells, but we're still trying to figure that out. So then to expand this into a human situation, looking at human myxomatous mitral valves, again, we see a little bit of phosphosmad2,3 and SMA and also snail in these control valves, but in the myxomatous valves, we see a strong induction, and there's a lot of activity going on at the surface of these valves. Again, the green is the SMA, so indicative of fibroblast activation. The pink is going to be phosphosmad2,3, indicative of TGF-beta signaling, and the snail is also expressed. We also see a lot of expression in the interstitial cells. And so there may be independent TGF-beta events in the interstitial cells, which has been associated with mitral valve disease in the past, and there may also be snail activity in both these surface cells and the interstitial cells. And where we think we see a lot of this activity is in areas where there's a lot of proteoglycan towards the surface of the valves. And that may be a matrix activation event that's involved in contributing to what we think is an endothelial mesenchymal transition in these human valves. So I've shown you a lot of descriptive data, but I think to really nail down if there's actually an endothelial mesenchymal transition, we have to do a lineage analysis. And so we've gone back to the osteogenesis imperfecta mice. This is a little bit complicated, but basically we use a genetic approach where we express TIE2, so TIE2 is an endothelial specific CRE in a transgenic mouse. And this is an estrogen receptor fusion, and so it will only get activated when the mice have been given tamoxifen. And we breed that to a reporter mouse, so if the CRE is active, we see beta-gal indicative of blue cells, and then we breed these two genes into either the wild type or the osteogenesis imperfecta valves, and we give them tamoxifen as adults. And so basically nothing happens in the CRE activity until you give them tamoxifen. We give them that at 4 months. they put them on that for a week and then look 21 days later. So if any endothelial mesenchymal transition has happened in those 3 weeks, we should see interstitial cells that are blue because they've come out of the endothelium and migrated into the interstitium. Whereas if there's no EMT, all of the cells are going to stay on the surface in the endothelial cells. And so the top ones are the control, the bottom ones are the osteogenesis imperfecta. And so in the control, you can see that basically all of the blue cells are in the, still in the endothelial layer, whereas in the osteogenesis imperfecta, we see interstitial cells in that thickened blue region of the valves. And so we think this is pretty good evidence that there's an endothelial mesenchymal transition happening in these adult mice that leads to new cells being formed and migrating into the valves. And again, we don't know if these are cells that are contributing to repair or disease or pathogenesis or any of that. And so we really need to do a lot more to figure out what these cells are doing. And this is just a quantification to show you that we see increased interstitial cells along the length of the valves. And again, this is some immunostaining basically showing in the osteogenesis imperfecta. So the green cells are the endothelial cells, the red cells are the beta-gal positive, and here you can see what we're thinking. We think we've caught these cells in the act of coming out of the endothelium and then going into the interstitium. And again, we think that this is pretty good evidence that there's EMT going on in these diseased valves. So to go back to development, we know that all of these players seem to be active in the adult valves. And again, this model is based on what we know is going on in endocardial cushions, but we think the same regulatory hierarchies are involved in the diseased valves. And again, this may be involved in shutting down adhesion, which is necessary for those cells to delaminate and migrate in, also involved in migration. And then these mesenchymal cells, we know, well, we think that they go in, and then we know that there's increased proteoglycan expression in those osteogenesis imperfecta valves. And so we're in the process of trying to figure out how this pathway regulates these processes in the diseased valves. Okay, so then to go on to try and look at some of the pathways involved in stratification of the valves. So the valves are stratified into elastin, proteoglycan, and collagen-rich layers. Normally, these layers are very well defined and very well controlled, but in disease, basically the matrix expands and is highly active. And this is a myxomatous valve, and you can see that there's just tremendous amounts of proteoglycan in these valves, and the organization has really fallen apart at this stage. So in trying to understand both the normal stratification and then some of the disease processes, we've looked to other connective tissue lineages. And so there's a lot of similarities between the spongiosa and cartilage in terms of expression of transcription factors and growth factors that control these lineages, and also in terms of the highly chondroitin-rich the chondroitin proteoglycans that are highly expressed here. In the fibrosa layer, we know that some aspects of very early bone formation are conserved, but certainly not the mineralization process in a normal valve. But in a diseased valve, a lot of these bone pathways do get activated. And so our thinking is that this fibrosa layer in an aortic valve, this is going to be on the side away from blood flow, which is where the calcific nodules usually form, that layer may be osteogenic. And so in thinking about myxomatous disease, we're thinking in terms of cartilage pathways, and then in terms of thinking of calcific disease, we think of osteogenic pathways. And then thinking in terms of some of the players that might be involved, we know that in the valve stratification, SOX9 is a critical transcription factor for cartilage, and then some of the matrix genes that are expressed in the stratified valves are these proteoglycans, elastins, and fibrillar collagen. And in thinking how these are regulated, again, we take lessons from cartilage and bone in terms of where we know SOX9 is involved in regulating extracellular matrix genes that are characteristic of cartilage. And then in bone, which we think is the calcific pathways, RUNX2 is a major transcription factor that then goes on to produce these mineralized proteins. And so what I'm going to focus on is the cartilage aspect of it in terms of the hierarchy of SOX9 and these collagen 2, collagen 10, and aggrecan, which are extracellular matrix proteins that are very characteristic of cartilage. And so when Joy Lincoln was a postdoc in the lab, she knocked out SOX9 in the valves, and these mice don't survive after birth because they have a lot of skeletal problems. But when you look in the valves, these are the control valves, these are the knockout valves. Basically, we see loss of proteoglycans when SOX9 is knocked out. We don't get any collagen 2, we don't get any cartilage link protein, which is also called hapalin 1. So basically, without SOX9, this whole cartilage regulatory program does not get active in the valves, and these mice do not survive. But interestingly, if you look at the heterozygous mice survive, but as adults, we see that the loss of SOX9, which again leads to loss of all the what we think are the a reduction probably of the proteoglycan component, we see increased calcification indicated in black here. We also see some thickening of the valves, both in the mitral and the semilunar position. So we think that, and Joy's gone on to show that loss of SOX9 that SOX9 is protective, and that loss of SOX9 leads to calcification in aortic valves. Okay, so what's upstream of this? The pathway that we've really focused on here is the Wnt beta-catenin pathway, and then going back into what happens during normal skeletal development. We know that in this zone of transition between cartilage and bone, the Wnt pathway is involved in repressing SOX9, so it inhibits cartilage formation, and it activates RUNX2, and it promotes bone formation. And so we wanted to see if this pathway is also conserved in valve development and then also in valve disease. So in looking at, is this pathway active during valve development and in adult valves, this is a beta-gout reporter where we see Wnt activity indicative of blue in the mitral valves and in the semilunar valves at late embryonic stages. And we also see Wnt staining in the adult valves, or the later valves, in sort of these little stochastic little bursts. And then axon 2 is a Wnt downstream target. We see expression again in the semilunar valves, sort of throughout the valves, and then we also see a reporter gene active in adult valves. And so what this tells us is that the Wnt pathway is active both during valve development and during adult valve homeostasis. So we wanted to figure out what this pathway was doing in the normal valves. So we looked in two different systems. So we did embryonic day 14 chicken embryo valves, and we cultured those as whole valves and then treated them with drugs. And so you can do either an activation or repression depending on which drug you give it. And then we also did in mice where we did a knockout of beta-catenin driven by a periostin Cree, which will affect the interstitial cells. So this is an example of the endocardial cushion, or the valve primordia cultures, where we take the aortic valve, basically the whole valve, cut it, dissect it out with the annulus region, and then culture it for about a week. And what you can see is that it maintains this matrix stratification. So in the Pentachrome stain, you can see elastin, proteoglycan, and collagen within these valve primordia. And then with the antibody staining, you can see that all locates out. And so when we treated these with the drugs, so with the Wnt activation, basically we lose the proteoglycan, but with the Wnt inhibition, we lose collagen. And so that again says that this Wnt pathway seems to be promoting the collagen and inhibiting the proteoglycan in these cultures, which is consistent with what we know from the bone experiments. And then when we look at this in more detail, if we look at the Wnt activation situation, we lose SOX9. So here you're looking at yellow. We lose aggrecan, we lose collagen. Whereas if we inhibit the Wnt pathway, the SOX9 is highly increased and it's nuclear. We get more aggrecan, we get more collagen. And so this is consistent with the Wnt pathway normally inhibiting these molecules. And when we inhibit the Wnt pathway, then all of this goes up. And so again, that suggests that the Wnt signaling pathway is inhibiting the chondrogenic gene program. So then we went into the mice to see what this pathway is doing in vivo. Basically, we have a conditional knockout for beta-catenin. We bred them in with a periostin Cree, and so that's giving us interstitial recombination in the semilunar and also the mitral valves. Notably, it's in the interstitial cells, but it's not in the endothelial cells. And this just shows that we were able to knock it out. So we see green is the beta-catenin. We normally see some expression here. Red is beta-catenin. But in the periostin Cree beta-catenin, we lost the interstitial beta-catenin, but the endothelial beta-catenin is still there. Axin 2, again, this Wnt signaling pathway indicator is down, and so we think that the knockout is working. In these postnatal mice, we do see less Wnt beta-catenin signaling in the valves. So the controls are on the left, the mutants are on the right, and what we see is over time that these mice with loss of beta-catenin in the interstitial cells develop these big proteoglycan-rich nodules that have sort of these huge hypertrophic cells in them. And this is reminiscent of what we think is going on in cartilage development with these hypertrophic cartilage cells. And so basically, with the loss of beta-catenin, they develop these over time. And we see these most predominantly in the aortic valves, a little bit in the pulmonic, but not very much in the AV valves. So then we looked at some of the gene expression, and we see that in the loss of function nodules that we see increased nuclear localization of SOX9. We also see a big induction of haplin 1 in those nodules. And then this is just a quantification of that. So in these nodules, we think that we're getting an increased cartilage gene expression program. And then there also seems to be a big switch in the proteoglycans. So normally the versican is the predominant proteoglycan in these valves. In the nodules, we don't see very much versican, but again, we see a lot of this cartilage link protein, and we see a huge induction of aggrecan, which is not normally supposed to be expressed in these valves. So again, we think this is an induction of the chondrogenic gene program. We see collagen 10, which is a collagen that's indicative of hypertrophic cartilage. Very specifically, in these nodules, we don't see any collagen 10 in a normal control valve. And again, collagen 2, which is in red here with aggrecan, is also induced. So we see a huge complement of cartilage-like extracellular matrix proteins that are activated in these mutant valves. And within the valves, there's very little of the fibrosa layer molecules. So looking at collagen 1, collagen 3, periostin is mostly excluded from the valves, the nodules, but there is upregulation in the thickened part of the valves. And so there's basically a dysregulation of all the matrix within the valves in the valves with the beta-catenin loss of function. So we think that what this is telling us is that within the context of an adult valve, so during homeostasis, Wnt beta-catenin signaling is necessary to limit the nuclear SOX9. And then when the beta-catenin is gone, all of this goes up and we see increased chondrogenic gene expression. There's other data out there that says that Wnt beta-catenin gain of function may be driving the osteogenic program and may be involved in valve calcification. And so it could be that normal valve is sort of sitting at a sort of Goldilocks point in terms of just right, where too much Wnt signaling can drive the valve to calcify, whereas too little can drive it to be myxomatous, and that may be involved in pathogenesis of both calcific disease and myxomatous disease. So what I hope I've convinced you of is that many of the regulatory hierarchies that are involved in early valve formation are also active in disease, and that regulatory pathways that control the matrix organization and homeostasis of the valve can also, when dysregulate, be involved in myxomatous or valve disease. And I've focused on myxomatous disease, but again, some of these pathways may also be involved in calcification as well. So I'll just stop there. Most of the people that did the work, Elaine Wierig is here. She's giving a talk later on calcification. Ming Feng did all of the beta-catenin work, and Christina and John also did some of the EMT work that I showed at the beginning, and I'll stop there.
Thank you for this beautiful lecture. I have just a couple of quick questions before we open for a discussion. And the first one is related to the endo-MT. So
yes.
I just wonder if you can speculate or you can rule out what's your thought about the activation of progenitor endothelial cell already in the leaflet that get activated and participate in the endo-EMT instead of like adult, like fully developed endothelial cell that transition to valve interstitial cell. If you think or you know of any indication that it could also be like a progenitor endothelial cell that some kind of activate to mimic actually the mechanism you see in the development. And the second one is just a curiosity on your beta-catenin loss of function and your knockout model. Do you see any effect on the root? Would that have any effect like on the mechanical function of the valve? Do they affect the structure of the aortic root?
So the first question is, we don't have a way of distinguishing between like an endothelial progenitor and a mature endothelial cell, because when we hit them with the Tie2, they're both going to be there. And I don't know if there's subpopulations of valve interstitial cells, some that have sort of a progenitor-like phenotype, and then others are more mature. And our sense is that when you take the valve interstitial cells out and you culture them, they all kind of do one thing on the collagen. And so they lose a lot of the information within the context of the valve. And so I don't have a great sense of the diversity of the interstitial cells within the context of the valve. But there may be different types of endothelial cells, and we haven't really parsed that out. As far as beta-catenin being some sort of structural mechanosensor, that's definitely a possibility. That's why we did the chicken experiments as well, because in that situation, we're hitting the pathway much lower down at the GSK complex, and we're not affecting the adhesion. So the fact that we get similar phenotypes when we do the XAV treatment in the cultures as we do when we do the beta-catenin knockout, we're thinking that it may be the intracellular functions of beta-catenin. But there certainly may be a mechanosensory function for beta-catenin that is involved. We are not affecting the endothelial cells. So, we're not really affecting the root cells with that periosteum Cree either. So there are some cells, adventitial cells, but not really the root is not really being recombined as much with that periosteum Cree. So there's definitely a possibility for beta-catenin involved in mechanosensory. It's not endothelial. We don't think it's the root. And then those drugs are recapitulating what we see with the beta-catenin knockout. So we think that it's the pathway, but we can't rule that out in any way.
Thank you very much. So the floor is open for discussion.
Yes.
Kishan Urin, cardiac surgeon from California.
Thank you.
Thank you very much. Kishan Urin from Calgary, Alberta, Canada. This is probably an obvious question, but have you looked at bicuspid I know you function in the mixing of these valves, and a lot of people, it's a separate topic these days. Have you looked at what's happening in bicuspid valves?
We have,
In relation to what you found?
yeah, we have not, and that it's limited a bit by the mouse models for bicuspid valve. I think there are some coming online that have more prevalent bicuspid aortic valve, but I think it would definitely be interesting to see if these pathways are also activated in the bicuspid aortic valve. Those have more of a propensity to calcify, I think. So the idea, but you know, it may be that you're looking in the opposite direction. Yeah. Well, and another interesting question is, how do you get 2 leaflets where you should have 3 leaflets? And so that's developmentally, that's an interesting question that I don't think anybody has the answer to that. So, yeah.
Thank you. Maybe one quick last question.
Sure. It's a great
Hey,
talk.
thanks.
And so, in human tissue, have you been able to get samples from, let's say, a sequestration dependent and a sequestration independent model of MMVD, right? So, if you look at, let's say, the Marfan's models, you know, the thoughts that the big issue there is lack of sequestration there's a lack of sequestration of TGF-beta in the matrix, right? And so, whereas most adults MMVDs or the garden variety MMVD may not be a sequestration-dependent pathway, and so, or mechanism. And so, is there a difference between? Have you been able to see whether there are different molecular signatures between those two?
Well, the ones that I showed are not Marfan's. And we've been looking at Marfan's, and there may be more TGF-beta, although the timing in the human is off. But we're also looking at the fibrillin mutant mice, and there we see, as has been reported, we see more TGF-beta. We're in the process of breeding those in with the Ty2 Cre-ER OIMs, hopefully this week. And then we'll see if there's increased EMT in those mice. We know there's increased snail. And there are areas in those valves that look activated, and there's phosphosmad 2,3. So, like I said, we're breeding those in now with the Ty2 Cre-ER in the reporter, and hopefully we'll see if. I think there's 2 different things going on. One is there's a matrix dysregulation that may be related to TGF, is almost certainly related to TGF-beta. In the osteogenesis imperfecta, actually, there is increased TGF-beta signaling in the bones, and that can be resolved with a TGF-beta blocking antibody. So what we're seeing in the osteogenesis mice may also be a TGF-beta related event that's associated with those fat proteoglycan-rich valves. I think there's an EMT issue, and I think there's a proteoglycan regulation issue, both going on at the same time. I'm not sure how they fit together.
All right, thank you very much.
Okay, thanks.
Lecture.